Okay, so this is just going to cover the phospholipid bilayer. So first I want to start by looking at the structure of just a little phospholipid here so we can understand why it works the way it works. So it has this phosphate group at the top with two fatty acid chains. They can either be two saturated fatty acid chains, or it could have an unsaturated fatty acid chain. And what the unsaturated is going to do is add this bend here. And this bend changes the properties a little bit of that phospholipid bilayer. Then we have two fatty acid chains, they're connected to a glycerol. Instead of making a triglyceride with three fatty acid chains, there's a phosphate group connected to the top. And this phosphate group is very polar, so the polar side likes water, making it hydrophilic, and the bottom, being very nonpolar with a bunch of CH bonds, makes it very hydrophobic. So the hydrophobic ends are going to want to move away from water, while the hydrophilic end is going to want to move towards water. So I'm going to try to draw it as best I can, but first we're going to look through this POGL we did in class to kind of understand what would happen. So water, I'm sorry for the bad picture, I'll try to redraw it here. But basically the idea was, if we had oil, how would the phospholipids attract around that? And what would happen is, and I'm going to draw the phosphate in red and the fatty acid tail in green. So the fatty acid tails would want to stick into or towards the hydrophobic oil. And then the hydrophilic tails would want to stick and go more towards the water. So this part being hydrophilic, just write HF, hydrophilic, and this side is hydrophobic. And what that does when you have a bunch is kind of like this picture's illustrating, it starts to form like a little barrier around it. So then what happens when we add these cells in pure water? Well, this won't happen. This situation with the phospholipid tails, if they're just in water, they won't want to form into tails like this. So if I drew it, I'm trying to give everyone two tails. If there was water in the middle and water on the outside, this is hydrophobic, so it's not going to want to be near the water, but even though these are hydrophilic. And vice versa too. If the tails were sticking in toward the water, because the heads were attracted to the water on the outside, once again this isn't going to work, because you got hydrophobic ends being around the water. So what's going to form is something like this, to where you have, and excuse the not drawing, but you're going to have a bilayer with all the tails in the middle, and then water in the middle and water on the outside. So you have the hydrophilic ends touching the water and the hydrophobic ends by each other, forming this double layer, or the phospholipid bilayer, because it's made of phospholipids and there's two layers. Okay, so let's talk about some things that are in this plasma, this phospholipid bilayer. One definite thing that can go in it is cholesterol here. And cholesterol kind of does the opposite of what a fatty acid chain will do. So this here is a drawing of a bent fatty acid chain. And so what that's going to cause is they're not going to be able to pack as tightly. If there's a bend in one, then they spread apart, and so the phospholipid bilayer becomes more fluid. Where if we add cholesterol, it's a hard thing. So if we add cholesterol to the phospholipid bilayer, it's going to become more rigid. So in the summer, your body may add more cholesterol to make the cells more rigid and stay together. But in the winter, when you don't want your cells to freeze, it'll take that cholesterol out and maybe even replace it with more saturated fatty acid chains, so that way it remains fluid. Also, some other things that can be found in the plasma membrane is proteins. And so all that I want you guys to know for now is that in a protein, when we look at the R groups, hydrophilic R groups will be most likely on a channel protein to be found on the hydrophilic sides of the plasma membrane, and the hydrophobic parts of a protein will be found in the hydrophobic part of the plasma membrane. So that's kind of how they stay oriented in the plasma membrane. I'll talk more about proteins when we get to transport. But you do have surface proteins, peripheral proteins, channel proteins. And then also attached to the phospholipid bilayer is glycoprotein. So we have a phospholipid bilayer here that I'm going to try to draw really fast. And basically what you do is you have these glycoproteins. So they can be stuck to proteins or not stuck to proteins. And they are just little, they're really carbohydrates. Oh, that's supposed to be a hexagon. They're really carbohydrates chained together, and they're your cell markers. So they help with cell communication. So just remember that there's proteins and cholesterol in a cell and how those work together. Thanks for watching.